My name is Dr. P.C. Patel, Dr. Kalyan Chandran sir, and Dr. M.S. Patel sir. They are the major guides in my Ph.D. study. So, response of foliar application of iron and sulfur in alleviation of iron chlorosis in acid lime, that is xylem. Iron chlorosis is a widespread physiological disorder which can have significant consequences for agricultural production. It is mostly observed in alkaline soils of arid semi-arid areas, as well as some very sandy soil having low organic matter. Even if sufficient total iron is present in the plant, it is not physiologically active. It could be controlled by foliar spray of ferrosulfate or iron chelate. Iron deficiency in soil and plant have been reported in many parts of India, including Gujarat. But at present, also, we are observing not only iron deficiency, but multi-nutrient deficiency, because the farmers are not using the farmyard manure, and they are using the indiscriminates of chemical fertilizers, high analysis fertilizers, and resulted in there will be a problem of the iron chlorosis in many crops, in sorghum, xylem, and other fruits also. Material methods. There were 4 levels of iron. There are 20 foliar treatments. So among there were 4 levels of iron: 0.05, 0.10, 0.15, 0.20% Fe as ferrosulfate without neutralization. That is IS1, IS2, IS3, and IS4. Another set of treatment with similar concentration were also taken with neutralization by calcium hydroxide. That is IS1N, IS2N, IS3N, and IS4N. Additional to treatment of iron with 0.05 and 0.10% FeS ferrosulfate mixed with 0.025 and 0.05% citric acid, respectively, were included. That is, the name of the treatment is IS1C and IS2C. The chelated form of iron from 2 different salts were used with 0.05 and 0.10% Fe. The name is IA1A and IE2A for ammonium salt of FeEDTA, and IE1S and I2S for sodium salt of FeEDTA. Another source we have taken ferrous chloride was third source of iron, in which 2 concentration of 0.05 and 0.10% FeS ferrichlorides were included, that is IC1 and IC2. In case of sulfur, only 2 levels, that is 0.05 and 0.10% as sulfur as calcium sulfate, CS1 and CS2 were kept. 2 controlled distilled water spray and no spray, that is absolute control in randomized block design. The foliar spray were applied thrice at an interval of 10 days. So we have kept 2 control. So distilled water spray and no spray control, that is absolute control. Site of the experiment. The site of the experiment at farmers' citrus orchard at Deesa, Banaskantha, Gujarat, India. The trial was conducted in 93-94 during my PG problem research work at C.P.College of Agriculture, Gujarat Agricultural University, Sardarkrushinagar, Gujarat, India. The soils were loamy and typical Pushti apartments, and soil have pH 7.6 to 7.9, electric conductivity 0.14, 0.16 decimol per meter, calcium carbonate 2.7, 4.8 gram per kg, calcium. Then extractable sulfur P2O5 24.3 to 25.4, DTB extractable ammonia Iron, manganese, zinc, and copper were 5.4 to 6.2, 18.4 to 24.0, 0.7 to 1.1, and 0.4 to 1.0 milligram per kg, means ppm, respectively. The irrigation water contained 342 ppm bicarbonate ion. Pressed leaves were collected on 8, 18, and 60 days after the first spray. Active ion from the pressed leaf sample was extracted by the standard method. So, treatments. So here, the application of ferrous sulfate along with the citric acid. So higher % decrease in chlorosis on individual period as well as on the average. And it was at par with ferrous sulfate 1% spray. So citrate is a natural carrier of ion in xylem transport. So citric acid mixed with ferrous sulfate and dissolved in water might have caused higher efficacy of IS2C and IS1C treatment. The treatments IS2C and IS4 and IS3, which have mainly iron content as ferrous sulfate, gave the higher fruit yield because of their higher efficiency in controlling the iron chlorosis. So these three treatments have controlled the iron more as compared to other treatments. So this treatment gave the higher fruit yield of the citrus, that is acid line. So here you can see this treatment, ferrous sulfate 0.5% plus citric acid. So I have taken this observation after spraying of this on 2 to 3 days. You can see here the recovery, it is a control plot. Here 0.25% ferrous sulfate, citric acid. So both the treatments control the iron chlorosis very effectively, and these two treatments give significantly higher fruit yield as compared to the other treatment. Effect of iron chlorosis. So I told that this treatment controls the effectively iron chlorosis. Then what are the causes of iron chlorosis? So leaf sap pH, then active ion. So the spray treatment of 0.5% ferrous sulfate along with the 0.05% citric acid solution IS2C showed significantly higher active ion content in leaves. Here you can see individual observation, period observation, and on the main basis also it showed higher active ion as compared to other treatments. Active ion content of leaf exhibited significant positive correlation with the % decrease in chlorosis. So as the active ion content increasing, the chlorosis has been controlled properly. So % decrease in chlorosis. So there is a positive significant correlation. There was a positive significant association of total ion with active ion. So when the total ion is more, then we are getting the higher active ion. So there is a positive correlation with total ion and active ion. The result indicate that all the ion treatment registered significantly higher active ion content along with increase in total ion content. Then third one is sulfur. Iron and sulfur treatment caused significant higher sulfur content in leaf than both the control. The soil was deficient in available sulfur, that is 7.30 ppm. In control 0.03, critical deficiency concentration in acid line for sulfur is 0.2%. The nutrient state of the control they can be stated as deficient, and increasing as content in the leaf due to sulfur and iron treatment would have directly contributed for the control of the iron chlorosis. When the efficacy of iron and S treatment, so these are iron and S treatments which recorded fairly higher S content in leaf is considered, it appears to help in activation of iron for chlorophyll synthesis and indicated earlier. So iron is responsible for the chlorophyll synthesis. Then total iron. Total iron content of the leaf samples average over 3 periods revealed that spray of 0.15% ferrous iron as ferrous sulfate IS3 show highest value 829 ppm, and it was similar with the spray of 0.20% Fe as FeSO4 with neutralization IS4N and without neutralization IS4 treatment. The total iron content on the basis of the pool analysis show a significantly high positive correlation with the percent decrease in chlorosis. So when the total iron content is increasing in the analysis, then there will be the percent decrease in chlorosis. Then effect on fruit yield. The spray of 0.05% iron as ferrous sulfate in 0.025% citric acid solution, that is IS1C and 0.10% iron as ammonium salt of FeDTA IE2A treatment were also similar with IS2C. So IS2C treatment so higher fruit yield as compared to the other treatment. So it gives significantly higher, followed by all the treatment ferrous sulfate, IS2, IS3, and IS4, and IS1 neutralization treatment also show lowest value. But IS2 neutralization, IS3 neutralization, IS4 neutralization, so higher, significantly higher fruit yield as compared to the control plot. So there was a positive effect of fruit yield of citrus due to sulfur application. So the percent increase in fruit yield varied from 63.1 to 94.4 due to iron treatment as compared with the control. So when we are controlling the iron chlorosis, then definitely we can increase the significantly fruit yield of citrus crop. So improvement of acid line fruit quality by foliar spraying of parasulfate. So here you can see the size of the lemon citrus as compared to the control treatment. So you can see the size. So here the size increases. Summary, the results suggested that the chlorosis in acid line may be attributed to higher soil pH, high bicarbonate in irrigation water, and frequent deficiency in soil and low active iron, total iron, sulfur content in chlorotic leaves. The spraying of 0.1% iron as ferrous sulfate, 0.05% citric acid solution, IS2C proved was more effective as compared with the treatment in controlling the iron chlorosis. Also, IS1C treatment having ferrous sulfate 0.25% and citric acid 0.025% foliar spray can control the chlorosis of the citrus trees. The total iron content of the leaf sample average over 3 periods revealed that spray of 0.15% iron as ferrous sulfate IS3 show the highest value of 829 ppm. It was similar with the spray of 0.20% iron as ferrous sulfate with neutralization IS4N and without neutralization IS4 treatment. The total iron content on the basis of the pool analysis show significantly high positive correlation 0.670 with the percent decrease in chlorosis. So suggestion, the results suggest that the spray of 0.1% iron as ferrous sulfate in 0.05% citric acid solution prove more effective compared to other treatments in controlling this iron chlorosis. Also, IS1C treatment having ferrous sulfate 0.25% and citric acid 0.025% foliar spray can control the chlorosis of the citrus tree. So at present, the farmers are facing the problems of iron chlorosis in citrus crop. So they can be advised to spray the ferrous sulfate along with the citric acid. So these treatments are more effective, and it also decrease the chlorosis and increase the fruit yield of the citrus crop. Thank you very much.